In this lecture, we're going to talk about Mendel's hereditary units, which we now call genes, and we're going to look at how these genes, or how the DNA in the gene, is used by the cell to tell it how to make proteins. So this is one of the basic fundamentals of biology. We call it the central dogma. And to put it briefly, central dogma, the cell uses DNA to make something called messenger RNA. They're both nucleic acids, but the messenger RNA is actually going to carry the message from the DNA, and it's going to carry it over to a ribosome where the proteins are then going to be made. So DNA to messenger RNA to proteins, that's the outline of the central dogma. And let's start with prokaryotes. Let's look at those first, like bacteria. In the central dogma is basically a two-step process, and here's the first one. So in the first step, DNA is used to make something called little m RNA, where little m stands for messenger RNA. I want to remind you that DNA and RNA are both nucleic acids. They're both made out of strings or strands of nucleotides, but in our last lecture we did go over some differences. Let's just review that briefly. So DNA, as you see in the picture, is double-stranded. RNA is single-stranded. RNA has uracil instead of thymine, but otherwise it has the other three bases: adenine, cytosine, and guanine. And then it has a different sugar, ribose instead of deoxyribose. And it has a different function. But they're both nucleic acids. So DNA is used to make messenger RNA. That's called transcription. And then that messenger RNA is used by the cell to make the polypeptide or protein. The information is used in the messenger RNA. That's translation. So DNA to messenger RNA is transcription. Messenger RNA to protein is translation. And in a prokaryote, that's occurring, in the cell. Remember, they don't have intracellular membrane-bound organelles, no nucleus. So this is all occurring inside the cell. And they do have ribosomes. So proteins, that's why they're showing you a ribosome here, because proteins are always made on ribosomes. We'll look at that in detail shortly. Now let's look at eukaryotic cells. It's quite similar with one extra step. In eukaryotes, transcription occurs in the nucleus of the cell. And instead of the messenger RNA, the finished messenger RNA being made right away, first there is what they call pre-mRNA that is made that has to then be processed. You have to cut out some pieces of it, splice the rest of it back together to form the finished messenger RNA. They call that RNA processing. So that happens in eukaryotic cells. But otherwise, it's similar. Transcription is going from DNA to making messenger RNA. That messenger RNA is now going to leave the nucleus, go out into cytoplasm, bind to a ribosome, and be translated into protein. So transcription followed by translation. So in a eukaryotic cell, you need to know that transcription is happening in the nucleus, translation in the cytoplasm. So let's look at transcription in a little more detail. So when you want to transcribe a gene, a piece of DNA that is, serving as a code for a particular protein, if you want to make a messenger RNA from that DNA, you have to do something similar that we saw in DNA replication. You have to separate the two strands. So we see that happening down here. The hydrogen bonds between the nitrogen bases are broken, and the two strands are going to unzip or separate, and you see a little piece of RNA is being synthesized here. And the enzyme that orchestrates this is called RNA polymerase. So it's somewhat similar to DNA polymerase. If you remember, that's the main enzyme involved in DNA replication. And like DNA polymerase, RNA polymerase always builds the messenger RNA in the 5' to 3' direction. So here's a close-up of this messenger RNA production or synthesis, transcription. So it's going to follow the good old base pair rule. If there's a thymine in the DNA, then when the cell makes the messenger RNA, it's going to put an adenine in the messenger RNA. And then remember, thymine is substituted with uracil in messenger RNA. So if there's an adenine in the DNA, you're going to put a uracil in the messenger RNA. Guanine nucleotide in the DNA, cytosine in the messenger RNA, and so on. So that's how the messenger RNA is made off of the DNA, and when it's finished, it detaches and leaves the nucleus, and then the DNA is going to zip back up together. So here's the double-stranded DNA that we used. We actually used this strand right here, so they call it the template strand. It's like the blueprint strand. The coding strand, they also sometimes call it. And look down here at the messenger RNA in red. You notice that the base pairs line up. Adenine in the DNA, uracil in the messenger RNA, cytosine, guanine, and so on. So here's our messenger RNA. And then let's look next at one of the most interesting parts of protein synthesis is how is it that with only 4 nucleotides in the messenger RNA, uracil, guanine, cytosine, and adenine, how is it with just with those 4 different kinds of nucleotides, messenger RNA can uniquely specify 20 different amino acids. There's 20 different amino acids in proteins, only 4 different kinds of nucleotides. So what we want to look at next is the code. How does the code work? Because that's what DNA and messenger RNA are. They're codes, they're recipes. They're not actually physically used to make the protein. Just like you wouldn't cut up your recipe for chocolate cake and put it in your cake. Or if you had a nice cookbook, which is sort of what the DNA is like, and you scribble your recipe on a piece of scrap paper, which is more like the messenger RNA, that too, you're not going to actually put that in your recipe. You're just using the information. DNA and RNA are information-containing molecules, and they guide the cell in making proteins. They tell it, take this amino acid and then add the second amino acid to it, and then the third amino acid would be, and it specifies which of the 20. Alright, so we're looking at how that works next. So you've got 4 different nucleotides, 20 amino acids. So it's pretty obvious that it's not going to work for each amino acid to have its own nucleotide. You know, like uracil will code for tryptophan and guanine for phenylalanine. So that's not going to work because there aren't enough. Now, what about every 2 codons? What about every 2 nucleotides? Because if you have 4 different nucleotides and you put them in various combinations of 2, like GG, UA, AU, CG, and so on, then you have 4 squared or 16 different unique kinds of combinations, starting with 4 nucleotides. So that's almost enough, but not enough. So that's not going to work either. So what about groups of 3 nucleotides? If you have 4 different nucleotides, how many combinations of 3 can you have? Like UUU, GGA, AGU, and so on. So 4 cubed, 4 times 4 times 4 is 64. So there's 64 unique 3 nucleotide combinations, and so that's more than enough, and that is, as it turns out, the way the genetic code, as we call it, works. It is a triplet code, meaning every 3 adjacent nucleotides in the messenger RNA is a code that tells the cell which amino acid to add next. They call it a codon. So a codon is 3 adjacent nucleotides in a messenger RNA, and it represents a very specific amino acid, like UGG. Anytime a cell sees that, it knows that the amino acid it needs to add is called tryptophan. Okay, and then what's it going to put next? Well, it looks at what are the next 3 nucleotides. UUU, what is that for? Phenylalanine. GGC, glycine. UCA, serine. And these get connected together by peptide bonds to form, the new protein. So that's how the genetic code works, this triplet code. Once again, it's the messenger RNA is made up of a series of codons, each codon made up of 3 nucleotides, and the sequence and the which nucleotides they are in the sequence has a special meaning for a specific amino acid. The genetic code is for the most part universal, which explains why they can take a piece of DNA from one organism and put it in another organism, and that organism can make protein from it. Because here it is. This is the genetic code. They've worked it out. They figured out what every possible all 64 possible codons, what they tell the cell to add or to do. So just to orient you here, these are the codons here, these letters, these triplet codes here. And then the amino acids are also 3 letters, like VAL for valine, arginine, over here serine. So those are the amino acids, tryptophan, TRP, glycine, GLY, and so on. So those are the amino acids. So for example, just to make sure you are with me here, let's look at valine for a minute. Valine is an amino acid. Notice it has 4 different codons that tell the cell to add valine next. GUU, GUC, GUA, GUG. All of those codons are for valine. Which, you have more than enough. There's, again, 20 amino acids, 64 possible codons. So you can have multiple codons for the same amino acid. In fact, they refer to the genetic code as redundant because not all, but most amino acids have at least 2 codons. And some have a lot, like look at leucine. Leucine's got 4, 5, 6 different codons for one amino acid. But then, like tryptophan only has 1 codon. So it's not evenly distributed, but this is the code, the genetic code, and again, it's for the most part universal. There are some exceptions. Archaea, there's a few codons that code for something else. I think mitochondria have some slight differences, but for the most part it's used by all organisms. Eukaryotes, plants, animals, bacteria, and so on. Notice while we're looking at this that some 3 of the codons are not for an amino acid. They tell the cell to stop, that it's finished. So UAA, UAG, and UGA are called stop codons. When you get to that, the cell knows the protein's finished and it releases it from the ribosome. Also, there is a start codon that also happens to be the codon for the amino acid methionine. So when you're making proteins, they all start with methionine. But if you looked at finished proteins in cells, a lot of them don't have a methionine at the start. because later it often gets chopped off. But initially it's going to be there. Okay, so this is just what I've already mentioned, spelled out. The genetic code is nearly universal, and because of that, genes can be transplanted from one species to another and can still be transcribed and translated into proteins. So you can put the gene to make the hormone insulin in bacteria, and they'll make the hormone insulin. So here we see some examples of where they took, for example, gene from a firefly that allows them to fluoresce, and they put it in a tobacco plant, or jellyfish genes in a pig. So again, this works because the genetic code is basically universal. Alright, this slide has too much text on it, but I want you to focus on these two vocabulary terms, introns and exons. So I do want you to know what these are. These relate to RNA splicing. So just to remind you, in eukaryotes, when DNA is transcribed into messenger RNA, you first make a pre-messenger RNA that you have to process. So here's where the terms intron and exon come in. Here's the pre-messenger RNA. This is a long chain of nucleotides, and you're going to have to cut out some parts, and the parts you cut out are called introns. So those have to be cut out, and then the exons remaining are spliced together to form the finished messenger RNA. Okay, so that's just a little more detail about pre-mRNA RNA processing. Introns are removed, exons are spliced together during RNA processing. This is for eukaryotes, not prokaryotes. Often when you look at the finished protein that's made off of from this messenger RNA, you'll see that the exons correspond to specific what they call domains or subunits in the proteins, where they may have different regions that have specific functions. For example, if this is an enzyme, perhaps domain 1 contains the active site, and domain 2 might be an allosteric site, or it might bind with a regulator or inhibitor, and maybe domain 3 would help anchor the protein in the cell membrane. So often these exons, even though they do get spliced together and translated into a single protein, they often do correlate with specific functional domains in the protein. Okay, so now we're going to look in more detail at how exactly is the messenger RNA translated into protein. How do you read the codons? How does the cell literally, physically bring the right amino acids over to the ribosome to make the protein? So, to answer this question, we have to talk about the importance of all three types of RNA. The messenger RNA, we've already talked about. It contains the code, the codons, the information. The ribosome, we've talked about before, is made of ribosomal RNA. If you recall, that's made in the nucleus and the nucleolus, and then transported out into the cytoplasm for eukaryotes. And the importance of ribosomal RNA or ribosomes is that you must have the ribosome because it is the site where the protein is made. It helps coordinate everything. It lines things up, and it makes translation possible. So even prokaryotes have ribosomes. Everybody has ribosomes. They're not membrane bound. They're organelles, but not membrane bound. So you got to have the ribosome. And then here's something we haven't talked about before. Let's look at this one right here. This is the other kind of RNA that's important. It's called little tRNA, little t for transfer RNA. And so transfer RNA is called that because it's the one that is going to transfer the right amino acid over to the ribosome to build the protein. So let's look at how it does that. So I'm going to use this same picture here. So look closely at this transfer RNA. It kind of looks like a firecracker, and on one end it's got an amino acid hanging attached to it, glycine in this case. And then on the other end it has 3 nucleotides lined up down there, and together they are what is called the anticodon. Okay, and it's called the anticodon because it is going to base pair with the codon of the messenger RNA. And they're going to follow the good old base pair rule. So if there's a G, a guanine nucleotide in the messenger RNA, then the corresponding anticodon nucleotide needs to be C, because C and G base pair. Here's another G, so another C in the anticodon, and then here's a cytosine in the messenger RNA codon, so this is going to be a guanine in the anticodon. So CCG will base pair with GGC, and they'll line up, and it'll allow this transfer RNA to sort of dock here, bringing in the amino acid to be attached next. Now, for this particular transfer RNA, it is only going to be carrying glycine. It's not going to be carrying any of the other 20 amino acids, because this is the transfer RNA that is carrying the amino acid that you need to bring over if the codon is GGC. So if you want to know what kind of amino acid would be on this transfer RNA, and you know that it's docking with GGC, the codon, you just look that up, GGC, on the genetic code. GGC is here, glycine. Notice GGC is the codon for glycine. So that's what the transfer RNA is carrying. So again, if you're trying to figure out if you know the anticodon and you want to figure out what amino acid is this transfer RNA carrying, don't look up the anticodon on the genetic code. Look up the codon. The genetic code is based on the codons, the messenger RNA codons. Alright, so let's look at the transfer RNA in a larger picture here. So this is the actual shape of it. It is a single-stranded nucleotide strand, but it is kind of folded up. It does have this 3-dimensional shape, and at one end is the anticodon, and at the other end is the amino acid attachment site. But they like to draw it in this form, this kind of firecracker form here. Okay, and once again, remember that the anticodon is going to bind with the codon in the messenger RNA. So if you've got a messenger RNA down here, what's the codon that it's going to bind to? Well, it would be U, U, C, because A and U, G and C. Remember that there's no T, there's no thymine in RNA. Okay, so we're going to kind of just go through the process of making a protein through the steps. So, first thing that's going to happen is you're going to make the messenger RNA in the nucleus by transcription. So that part is not shown in this figure. That's already happened. But now we're looking at where does that messenger RNA go when it comes out into the cytoplasm. So it actually is going to come out of the nucleus in a eukaryote and attach to the small ribosomal subunit. Ribosomes actually have a big and a small subunit, and initially they're not together. So really the messenger RNA is initially binding to just the small subunit. So it's attached to it, and then the first thing that's going to bind to it is the transfer RNA that binds with the start codon. So up here in this little inset is a blow up of this site here, where you can see that the start codon if you look on the genetic code table, it's AUG. That's the start codon. It's also the codon for the amino acid methionine. So this transfer RNA is called the initiator transfer RNA. It's carrying methionine. It's got an anticodon that binds with the start codon. So this is sort of like step one of translation. Once that happens, then the large ribosomal subunit docks on the small ribosomal subunit, and when it does, it sort of locks the ribosome in place and provides 3 specific sites that are going to be involved in making the protein. They call it the E, the P, this is the P site, and the A site, EPA sites. And notice that when the large ribosomal subunit binds, the initiator transfer RNA with the methionine on it is now positioned in what they call the P site. Okay, now I'm going to show you this cycle of events that occur when a protein is made. So in this picture, the protein has already added a couple amino acids, which we didn't see on the previous picture. But let's start up here. Here we have a transfer RNA in the P site, and it's got several amino acids already attached to it. And now you want to add another one. So here's what happens. The next amino acid specified by this next codon here on the messenger RNA, that next amino acid that needs to be added is going to come in on a transfer RNA. Here it is. There's the amino acid. And then on the other end is the anticodon that's going to bind to the messenger RNA codon. So that's going to come into the A site. So here we see it down here docked in the A site. So now we've got 2 transfer RNAs. One with the peptide so far that has been formed, and the other side, the A site, with the next amino acid that you want to add in place. The next thing that happens here is that all the amino acids are shifted from the P site to the A site, and now the transfer RNA in the P site is empty. Okay, and then those are going to both just shift to the left, and the empty transfer RNA without its amino acid is going to be it's going to go into the E site, and then it's going to be sort of ejected. So I think of E as exit or eject. So it's going to leave, and then it's actually going to be reloaded with the correct amino acid that is specified by its anticodon. So you'll reload it and use it over and over again. But for now, it's kicked off the ribosome, and now we have, you know, the polypeptide chain with an extra another amino acid added to it, and then you can add yet another one. So a new amino acid can come into this A site, and the cycle goes round and round. They call this the elongation cycle. Now, one messenger RNA can be used to make many copies of the same protein. It's not like you can only make one protein off of the messenger RNA. And in fact, well, we'll talk about polyribosomes in a moment, but just to finish up with the making of the protein, you keep adding amino acids until you get to a stop codon, one of these 3 stop codons. And when a stop codon is reached, then instead of another transfer RNA coming in with the next amino acid, a release factor binds, and that's when the cell knows it's all done. The protein gets released. There it is. The ribosome sort of falls apart back into the big and the small subunit. The messenger RNA detaches. It's probably soon going to be degraded, and that protein is finished now. Now you can use the same messenger RNA to translate it with multiple ribosomes. This is called polyribosomes. We have multiple ribosomes translating the same piece of messenger RNA at the same time. They're just spaced along the messenger RNA. So that makes it more efficient for making lots of copies of the protein before the messenger RNA is degraded, because the messenger RNA does not last too long. Okay, so now that you understand the importance of the sequence of the nucleotides in the DNA, because that specifies the sequence of nucleotides in the messenger RNA, and then that spells out the codon, so that determines which amino acids are going to be added to make the protein, you can better now, I think, appreciate mutations, how mutations in the DNA can, have profound effects on the cell because they result in different proteins being made. Because cells will faithfully follow whatever the messenger RNA is telling them to add. So just like if you change the recipe from add a teaspoon of salt to add a tablespoon of salt, the cell's going to do that, and the recipe is not going to be the same. So that's analogous to what happens when the DNA is altered. When you have mutations, then it's going to alter the protein. Point mutations are when you just change one single base pair of a gene, and sometimes just one single point mutation can lead to abnormal proteins that have actual health consequences. So let's look at an example of that. Hemoglobin and sickle cell anemia. So hemoglobin is a protein in red blood cells. It carries oxygen for us, and your red blood cells are basically bags of hemoglobin, picking up and delivering oxygen. It's a fairly big gene. It's got many nucleotides in it, but we're only going to look at the relevant region where the change is in sickle cell. So we're just going to look at this one region here of the gene for hemoglobin. And in the normal, wild type means normal, in the normal hemoglobin, let's look at the 3' strand. The sequence of nucleotides is CTT. Okay, now let's look at this point mutation over here. What happens if you change the middle one from a thymine to an adenine? So instead of CTT, it's CAT. Well, let's see what effect that has. So normally, when you're using the template strand to make messenger RNA, the codon would be, if there's a C in the DNA, it would be G. T in the DNA, there'd be A and another A. So it'd be GAA would be the codon. This is a codon, right? And if you look that up on the genetic table, genetic code table, that corresponds to glutamine, the amino acid. If you change that middle nucleotide, then the codon becomes GUA instead of GAA, and that codes for a different amino acid called valine. And it just so happens that glutamine and valine are quite different. They have different properties. Valine is hydrophobic, glutamine is not. It causes the protein to fold up differently, because remember back when we talked about protein structure, we emphasized how the sequence of amino acids in a protein dictate how it folds up, its overall shape, and then we discussed how the shape of the protein is tied to its function. So if you change the sequence, the primary sequence, you might, and in this case you will, affect the structure and thus the function. So here we see, even on a cellular level, you can see the effects of that single mutation in the shape of red blood cells. Here's a normal red blood cell, which has about 250 million molecules of hemoglobin in it. And you see it's kind of roundish, they call it a biconcave disc, kind of sunken in the middle. here's a cell, a red blood cell with the sickled hemoglobin in it, the abnormal hemoglobin. So you see it has a different shape. It's shaped like a sickle or a crescent moon. And that's going to affect its ability to transport oxygen. So just that one mutation can lead to an abnormality in the protein that can have health effects. So in general, all kinds of mutations can occur that result in abnormal proteins. You could have point mutations. What if you insert an extra nucleotide in the DNA, or there'd be an extra one in the messenger RNA, or if you take one out, that can change the whole, all the codons, and thus the amino acids in the protein. So, so the main point is that changing the sequence of the nucleotides in the DNA leads to changes in the messenger RNA, which often leads to changes in the protein. Now, because the genetic code is redundant, because some amino acids have multiple codons, there is such a thing as a silent mutation where the DNA does get changed, but it just so happens that the new codon is still for the same amino acid. So the protein that's made is really not any different. It has the same amino acid where it should have been. It's just with a different version of the codon for that amino acid. That's called a silent mutation. It does not result in a change in the protein that's made. Okay, so after you review or after you study this material, I encourage you to spend some time looking at this summary slide, because it's got a lot in it, and it's all stuff you need to know. So it starts up here in the nucleus. This is a eukaryotic cell. It starts up here in the nucleus with transcription. So here they're showing you, RNA polymerase making the pre-mRNA that you then have to process by cutting out the introns. And then that messenger RNA comes out into the cytoplasm, binds to the ribosome. Everything gets positioned with the E, the P, and the A sites, and then the loaded transfer RNAs with their anticodons on one end and their amino acids on the other end are going to transfer or bring the correct amino acids over to build the new protein, all specified by the codons, the triplet code in the messenger RNA, which really in turn is just carried from the DNA itself. The DNA is ultimately carrying the information, and the messenger RNA is a temporary molecule made off the template strand. So that completes our lecture of protein synthesis, the central dogma.